welcome everybody to the Boyer Award for Postdoctoral Excellence. It's my pleasure to award it this year to Paige Henning, but I'm going to just say a couple quick things about Paul Boyer first. Paul Boyer was a graduate student here, got his PhD in 1943. And as far as I know, he was the only one of our graduate students in biochemistry to go on and win a Nobel Prize. Hopefully some of you will change that to more. But he then donated some of that money from the prize to the department. One of the things that it was used for was to create this, this award in Paul Boyer's name. And so it's a real pleasure to be able to give the certificate for the award today to Paige, and it's suitable for framing. So there you go. Now, Paige has done some great work, but I'm going to let Mike Sussman, her mentor, introduce that work.
Is this really on? I have my backup hearing aids in, so never mind. Anyway, so next slide. Oh, I do.
Yeah, you do it.
I am really happy to talk about Paige, introduce her. This is her ontogeny. Fancy word, she taught me. Her development. Started at Washington State University for a bachelor's, and then did her thesis at Washington State. What?
Speak into the mic.
Oh, I should speak into it. That's good. Now you can hear me. Okay. So she did her bachelor's at Washington State, and then she did a PhD, and her thesis was really cool. She was part of a group that sequenced the genome of Tenera subulata, and that out of that came a new peptide hormone that we know nothing about and that she'll talk about. Can you hear me now? Usually my voice is loud enough, but not anymore. I'm getting old. Oh, for the recording. Oh, okay. All right. Then she got a 3-year NSF graduate fellowship to come to my lab and study the SPH peptide hormone receptor protein in both subulata and Arabidopsis. It's a large peptide hormone family. I don't want to take her thunder, so I'm not going to say anything about it. Talking more about Paige, she is both meticulous and creative. And these are important traits of great scientists can be very difficult to juggle. And she's done an amazing job of that, and I'm sure will continue. Another particular is that she's very brave. Some of you who know me well would argue just coming to my lab demonstrates braveness. But in fact, she did more than that. She actually switched fields. She hadn't done much protein work, and she switched from developmental biology to protein chemistry in my lab. And has a great sense of humor, has even come up with some new bad jokes that I don't know if she'll be sharing them with you. Oh, so to summarize, she came to my lab 4 years ago with that fellowship. Her goals were to learn protein chemistry, number one. Number two was to find the SPH receptor. And number three was to teach me about flowers and genetics. She's made real nice progress on 1 and 2. Which, while not perfect or complete, her experiments suggest that the SPH peptide receptor is not one of the usual players. In other words, it's not a protein kinase or G protein coupled receptor. And indeed, like Mark Estelle, some of you may know, who's the auxin receptor and also a poppy SPH receptor, it may be a new unexpected paradigm for plant peptide hormone action. Goal number 3, in other words, teaching me, has turned out to be more difficult, I think, than she expected. And here she is starting. We went to lunch together, and she said, "Mike, this here is a flower." Wow. And then she told me she wants to come to my lab to answer the deepest possible molecular level questions that Darwin himself pondered. In other words, what controls the length of male and female sexual organs in flowers? And also, how am I going to get a job? So I said, I can do that. I'll help you with both of those. And I am so pleased that she joined my lab, and I hope she feels the same. And this is another experiment she recently completed. A beautiful baby named Juniper. And so that's it. Oh no, there's one more slide. Now, without further ado, I'd like to introduce this year's Boyer Postdoctoral Award, Dr. Paige Henning.
Oh, the seminar's just getting started. Don't leave yet. Okay, hypothetically, this microphone is picking me up because I turned it on. Okay, hi, as you all heard, my name is Paige Henning. I'm a postdoc in the Sussman Lab, and today I'm going to talk to you about my favorite topic, sex, specifically plant reproduction. That first part of the sentence was to capture your attention. I saw some head lifts, yeah. The second half was to set your expectations low. And that explanation was to let you know that I am that guy who gets in front of people and I try to make jokes when giving a serious presentation. Okay, great. Happy for that. So I'm sure when you thought about sex and reproduction, you thought about flowers, right? And there's a reason for that. So of the approximate 382,000 plant species, approximately 87% of them are flowering plants. Which brings up this question: how have angiosperms managed to dominate the plant world, the plant kingdom, right? Angiosperm, they're the only plant lineage to actually exist on every continent. So you might think Antarctica, surely there's no plants there. You'd be wrong. There are grasses there. So, like I said, how have angiosperms managed to dominate? The answer is the evolution of the flower and the fruit, which is why we're here today, to talk about flowers, because flowers are pretty and I don't care about fruit. Okay. As long as Mike laughs, that's all that matters. Everyone else, you can pity laugh as well. That'd be real great. So if we're going to talk about reproduction in terms of plants, we're also going to have to know words properly. And everyone in here probably isn't a plant biologist, and everyone in here probably doesn't know floral anatomy. So let's speed run this. Okay, so here in red, we have the anther. This is where our pollen is produced. Attached to the anther, we have our filament. Together, they make up the stamen. We then have our stigma. This is where our anther or our pollen falls. It falls onto the stigma, and then a pollen tube grows through the style, eventually reaching an ovary and fertilizing an egg. Here's a picture of a Arabidopsis flower. I've highlighted the anther here. Well, I forgot I have a pointer. I've highlighted the anther here in red. We can see that it actually has multiple anthers. And there's our little filament in blue. We have multiple filaments. And here we have our stigma, and then we have our style. So just to recap why you should care about any of this, our male gametes are produced in the anther with our pollen. Our pollen contains our male gametes. And then within the style somewhere, we have our ovary, which is where our eggs exist, or our female gametes. So I'm specifically interested in distyly, right? There are so many plant reproductive systems, you gotta pick one. And I picked distyly. It's a form of heterostyly, and it's really cool because a species will have individuals that either have S-morph flowers or L-morph flowers. So the S just stands for short style morph. So you can see our style is short in this individual, and our anther is placed high in the flower because we have this long filament. And then we have our L-morph individual, and that just stands for, you guessed it, long style. So yeah, we have our long style, and then we have our short filament phenotype. This is comparable to animals. At least that's what I tell people to make it easier for them. So you can either be an S-morph individual or an L-morph individual. You can't be both. Just like a human can be a male or female, they can't be both biologically. So distyly is super cool because it's a case of convergent evolution. It's a found in at least 28 families and 187 genera, with like a ton of different like points of evolution. Like it's been involved at tons of different times. And even within genre, for example, the linum, it's evolved multiple times in linum. And is of economic importance, hypothetically, because jasmine, starfruit, and buckwheat are all distylists. And we do eat starfruit, and we do eat buckwheat, and jasmine can be used in perfumes. So you should care at the very least because you enjoy eating. Hypothetically, maybe you don't. There are 5 general key characteristics of a distylist plant. So as we talked about, these anther height dimorphisms, where in one morph we have anthers higher in the flower than the other. And then our pistil length dimorphisms. So we have our stigma lower in one morph than the other. We have differences in our pollen grains. So hypothetically, even if you're not a plant biologist, you can see that there are clear differences between the pollen grain in figure A and in F. We have differences in our stigmatic papial. So hypothetically, even if you're not a plant biologist, that is very obvious in our figures B and E. And it's hypothesized that these dimorphisms evolved to discourage self-pollen from sticking to self-stigma. And then occasionally we have self-incompatibility. So that's an individual's inability to reproduce with itself. So a lot of plants, they can just mate with themselves and they don't need anyone else. Self-incompatibility just means that you can't do that. Sometimes we also see intramorph incompatibility, and that's when an S-morph individual is incapable of reproducing with an S-morph or vice versa. So there are a lot of distylist species that are studied at the genetic level, and I spent my PhD studying distyly in Passifloraceae, specifically in Turnera. So these are pictures of Turnera subulata. One thing Mike was right about is that you can't teach him how to pronounce things. I've been telling him it's Turnera this whole time. That wasn't, I don't care that he doesn't know how to pronounce it. That was a joke. I'm sorry, Mike. That was a joke, but okay, I'm moving on. So here's a picture of Turnera subulata again. It's beautiful. It's a subtropical shrub. Distyly is an ancestral trait, meaning the last common ancestor had distyly. So any species within the genus that is distylus is going to hypothetically have the same genes controlling that. It has self and intramorph incompatibility, which makes genetics easier. It has an annotated genome, which makes it an exceptional model to study in because we like to have genomes. And I annotated it, not to like right. And it has 3 S genes. So it's the genetic basis of distyly is relatively simple in Turnera compared to other distylus species. So we talked about S genes like for 1 second on that last slide. Let's dive deeper into the genetics of distyly. So yeah, I'm sure you guys all came here for a plant talk and the biochemistry award. You're welcome. So the genetic basis of distyly in all distylus plants is called the S locus, or the self-incompatibility locus. To date, it's always a hemizygous supergene. So supergene is just multiple loci that are inherited in a Mendelian fashion as if there are only one gene. To date, all of the distylus species that have been studied, the supergene is S-morph specific. So this means there's one copy, if it is a diploid individual, there's one copy of the S locus, and then the other chromosome would not have anything. There's no allelic region. In terms of Turnera specifically, we have our female mating type and morphology gene. We have a male mating type gene. So mating type just refers to who you're capable of mating with, right? So if you're an S-morph individual, your mating type means that you're capable of only breeding with L-morph and vice versa. And then we have our male morphology gene. So that's involved in establishing those filament dimorphisms. So our female gene we found was this acyltransferase that's involved in brassisteroid inactivation. We called it BAD after the name of the superfamily it's from. It's what causes that short style phenotype and establishes female self-incompatibility. We have YUC6. It's a member of the yucca family, which is involved in auxin biosynthesis through the tryptophan dependent pathway. It's the most bioactive pathway for auxin biosynthesis. It establishes that male mating type. And then we have SPH1, which is a member of the S protein homolog family. It's a family of cysteine-rich peptide hormones, and it's what contributes to that long filament phenotype. Now I'm going to highlight this because, as you may know, there are citations for these other two genes, and that's because I characterized those during my PhD, or I helped characterize them. The SPH1, however, eluded me because there's only so much time and only so many resources, and so this guy kind of got left out. So I made it my postdoc mission to characterize it. This is my mission. Okay. So let's talk about the S protein homolog family, just in general, just background knowledge. So like I said, it's a family of cysteine-rich peptide hormones, and there are two members with known functions. So as you might have guessed, there's SPH1. I'm going to specifically refer to the copy from Turnera subulata, so TSSPH1. It's from Dicylus turnera. It's involved in filament dimorphisms, and how we know that is through this little mutant right here. So here we have our wild type subulata, S-morph, wild type L-morph, and then our little mutant. This mutant is an S-morph individual, and you will notice it has short filaments, causing its anthers to be at the same level as the stigma. When we looked at expression of the 3 S genes, so BAD, YUC, and SPH, in this individual, we found that SPH was not expressed, and based off of that, we decided that's hypothetically our filament dimorphism gene. This was back in the paper was published in 2019, so you imagine a lot of work has come out since then. Yeah. The other member of the SPH family with a known function comes from poppy, so Papaver. So it's this PRSS. It's the founding member of the SPH family. It just stands for Papaver stigma S protein. It's involved in self-incompatibility, and it's an allelic system, which makes it really confusing and difficult to talk about. Basically, an individual will have their little S locus, the poppy individual, and that S locus will contain a copy of PRSS and its receptor, PRPS. And you're basically wanting to mate with someone who has a different copy of both of those genes from you, right? So hypothetically, if you mate with yourself, let's say this is us, our SPH, PRSS is secreted from our stigma. And then because it's our own receptor, we're going to be able, our pollen tube is going to perceive our self SPH, right? And it's going to cause a response within the pollen tube. It causes death. It causes death of the pollen tube. If we breed with someone else who has different alleles for these two proteins, when we secrete our PRSS or SPH from our stigma, it's not going to be perceived by the pollen tube specific receptor because it does not fit, hypothetically. That's what this diagram is showing. And that's going to allow for the pollen tube to continue elongating through the stigma into the style and eventually the ovary, and then it will fertilize the egg. So that's like our crash course on self-incompatibility in poppy. It really does not matter beyond that PRSS is the founding member. And another thing that's extremely important to know is that our receptor, PRPS, is Papaver specific. So PRPS does not exist outside of Papaver, and it really only exists in Papaver and then a very closely related genus that I cannot remember off of the top of my head, and I didn't write down, so I failed you if you were interested in that. But that was the wrong reason to come to this talk, if that was your point. Okay. So now we know about our two SPHs. Let's compare notes. So we have our Turnera subulata SPH1. We know that's involved in filament elongation. And while I didn't have a slide about this, I did search the Turnera subulata genome, and there are 77 family members in subulata. Hypothetically, there could be more because our genome is only at the scaffold level. We're not fancy. We're not at the chromosome level. In poppy, our poppy PRSS, our founding member, it's involved in self-incompatibility. It does this through programmed cell death, and it does have a receptor that is poppy specific. The one overlap that we see between our two proteins, our two peptide hormones, is that they're involved in reproduction because self-incompatibility and filaments. Yes. So this is a diagram that summarizes everything Franklin Tong's group has found about distyling or about self-incompatibility in poppy. And it's really just a lot. And I'm not going to go through all of it, except for I kind of am. But if you want a really detailed version, you can see this review, which is where I stole this from. So basically, what the poppy people know is that when we have perception of the SPH, PRSS, by its pollen tube specific receptor, PRPS, we see an increase in cytosolic calcium and a decrease in pH of the cytosol. And then we see increase in ROS and decrease of ATP, all within the pollen tube. So this is in the pollen tube. We have this inhibition of endocytosis. We have inhibition of this SPPase, which is a pyrophosphatase. This protein here, MPK9, it triggers programmed cell death and is involved in actin alterations. This protein here, PGAP, is involved in deacylation of inositols. They think hypothetically this might be the result of AHA inactivation. There's actually like a paper this year about mitochondria and like related to this, but that's not in the diagram, so we're not going to talk about that. But if you're eager to know about that, exists. And they hypothesize that deacylation of inositols is probably critical for SI response in poppy. And they think hypothetically, based off of their data, but they had nothing to prove this or like support this hypothesis, that there might be a GPI anchored protein that's acting as a co-receptor with PRPS. And I only bring all of this up because it's going to be cool in like 45 minutes if you can stay awake. Not 45, that would be excessive, but whatever. So we've increased what we know about PRSS. Obviously, it inhibits endocytosis, it increases ROS and calcium levels, decreases ATP, that's a typo, and cytosolic pH. There might be a GPI anchor protein co-receptor. Yeah, so we know a lot about poppy, don't know that much about SPH. And now you might be asking, what about our favorite plant ever, Arabidopsis thaliana? Well, not much is known about Arabidopsis or SPHs in Arabidopsis thaliana. We know that there are 98 family members, and we have the NMR structure for SPH15. One thing that we do know based off of this analysis in general is that the best way to determine if a peptide is an SPH isn't actually sequenced because they are very divergent in their sequence, but rather the number of cysteine residues they have and the number of beta sheets. So secondary structure is more important for determining if it's an SPH or not. What I have found through just downloading RNA-seq data and then being a nerd with my computer, you can laugh at that. God. Okay, is that apparently the SPHs, they're only expressed in reproductive tissue. So this is just RNA-seq data that I've turned into a heat map for you. And I've artificially set the cap of expression level. And we can see that all of our SPHs are only expressed in reproductive tissue. We don't see any expression in vegetative tissue. And if we look specifically at just like I've summarized it here for you, we can see again that we have very high expression of these in the SPHs in the stamen and the pollen. If I artificially cap it again, we see that we do actually have expression in the inflorescence apical meristem and then also during seed development. If we look at the specific family members in only reproductive tissue, we can see that we actually do have expression in a bunch of different reproductive tissue. Yeah, so overall, this kind of suggests that in Arabidopsis, we're also seeing that the SPHs are important for reproduction. We don't know how, this is just based off of RNA-seq data. For the Arabidopsis SPHs, like I said, we have that structure for SPH15, and there are 98 members. Based off of the number of members in Arabidopsis, the number of members in Turnera, and then I did a very large phylogeny of the family back in like during my PhD sometime, so I don't know whenever that was. This is true across a bunch of different genres, so the SPH family is massive. That's all. So now let's actually talk about cool experiments I've done and not just things I've downloaded or papers I've read or, yeah. So the question is, for me at least, because I'm interested in distally, is how is SPH1 eliciting filament elongation? And so the concept that I came here with was to do a multi-omic analysis of filament development. And when I came here, I was very naive in the world of biochemistry, and I did not realize how hard biochemistry was. Mike slapped me. So, yeah, and I still don't like Arabidopsis, but I'll use it. Okay, yeah. So the concept here is that I'd take the filaments from the S-more, do some RNA-seq, and then we'd make a co-expression network using that RNA-seq. One thing to consider is that really this lab is the only lab that's doing anything right now with Turner at a molecular level. And so there really aren't that many resources available for Turner outside of what I've done. Yeah, what I've done. I'm gonna take the claim for that. So yeah, we needed to generate some RNA-seq because RNA-seq just did not exist for the filaments, only the stamen. And then we decided it's time to enter the world of proteomics with Distyle and Turner. So we did some protein isolation. The idea here is that once we had these isolated proteins, we'd do a phospho enrichment so that we could look at phosphorylated peptides and differences in phosphorylation between the S and the L morph. And then we could take our non-phosphorylated peptides and just look at differences in protein abundance. And this is under the hypothesis, if we remember, SPH1 is hemizygous and S-morph specific. So only the S-morph has a genomic copy of SPH1. The L-morph does not, which is beautiful because we don't have to make knockouts or anything. We can just hypothesize that there are going to be differences due to SPH1 because it just only exists in one genotype, right? Yeah. Yeah. Okay. So then once we have all these differences and this co-expression network, we could combine them to create a final network. We could look within that final network to find clusters that contain SPH1, and then we could make a model for filament elongation. And so here I'm just showing you this so that we have a picture to look at while I give you numbers. So I found a cluster that had SPH1. It consisted of 165 different genes. Of the genes that were expressed, so the 8 different RNAs that we found, 67% of them had peptides, and then 59% of them had phosphorylated peptides, which are denoted by the circles versus like squares and yellow versus blue. When we look at Go terms, because everyone loves some Go terms, we found that reproductive structure development was one of our top terms, binding was a top term, and membrane-bound organelles was a top term, which is exciting because, again, we're looking at filaments in the flower. So I was hyped. I'm sure Mike was hyped too, even though he hates Go terms. Okay, so next what I did is, sorry. I'm so sorry. Okay, so next what I did is I took those proteins that had phosphorylation events that were specific to one morph or occurred more in one morph than the other. And I compared it to a database of Arabidopsis phosphorylation events. So it shows every single site on a protein that has been previously documented as capable of being phosphorylated. I don't remember the name of that database, but you can read about it here. Or I guess email me. So I did that, and I came up with this list of proteins that have previously identified phosphorylation sites. Specifically, these 5 are the ones that have the previously identified phosphorylation sites. You see that this one is interesting because it's only phosphorylated in the L morph, so the morph that lacks SPH. The rest of them are phosphorylated only in the S morph, or there are some phosphosites that occur at higher rates than in the L morph. So if we look at the actual roles of these proteins based off of their Arabidopsis homologs, so I assigned all Turner proteins Arabidopsis homologs so that we could have some idea of what a function might be. We see that these 3 proteins are involved in RNA processing, which is pretty cool. We see that this AT5 PTase 12 is a type 2 inositol phosphate 5-phosphatase, which is cool because that means it's involved in phosphoinositide. Whoops. Oh, and then I forgot that I didn't put description here because this is just an ethylene protein. So it's just a protein that's involved in ethylene response. But that was pretty exciting because if you remember, the poppy people were talking about phosphoinositides having a role potentially in self-incompatibility response based off of the role of that one protein. So that suggests that PIs likely are common between TSSPH1 and PRSS signaling. What I also found interesting in this analysis was the presence of this ATM1. So this is a kinase that promotes fertility, meiosis, mitosis, and it also prevents double-stranded breaks from occurring during crossing over during the endoreduplication cycle. The CDKG2, also interesting because it promotes endoreduplication and growth, cell growth expansion. So the endoreduplication cycle is just when a cell is going through mitosis, it may prematurely stop after the genome has duplicated, and it enters into the endoreduplication cycle, which just allows the cell to become much larger than it normally would, doubles in size. Plants use this cycle as a way to rapidly grow tissue, like filaments, hypothetically. So this then brought up the hypothesis, is TSSPH1 regulating the endoreduplication cycle? So as you might remember, I assigned Arabidopsis homologs to all of my Turner proteins, and that allows me to take advantage of tools like String, which mine PubMed for experiments and like actual support for interactions between proteins. Interactions is used loosely because like they might not actually be interacting. It might be that this is a homolog of this protein and this is a homolog of this protein, and they do something together. But anyways, it allows me to draw more conclusions from my data set than I might be able to if I only had the subulata names. And so by throwing all of my proteins into a string, I was able to come up with this little network and then a model for hypothetically how filaments are elongating in the L versus the S morph. And so the hypothesis here, we'll go through it on the L morph. Yellow means that the protein was found in both morphs, but not differentially expressed, so same level of abundance. White means that it was only found in the RNA-seq data. We didn't have peptides in our mass spec data set. And then this purple means that the protein was only found in the S morph individual. Our dark green means that the phosphosite only existed in the S morph, and then our light green means that it existed at a higher level in the S morph relative to the L. Okay, now that I've taught you about colors, let's go through this diagram. So because ATM is not phosphorylated in the L morph, we could probably assume that it's inactive. And if it's inactive, its SOG1 is likely inactive because SOG1 is activated by ATM1. If this is the case, then that means the CDKB1 is active, which forms a complex with this psych A34. Psych A34, it might be active as it is phosphorylated in both data sets. This complex together actually phosphorylates CDKG2 at a site that causes its degradation, so it gets marked for degradation. Hypothetically, since CDKG2 is not present in the L morph, but it is in the S morph, we might hypothesize that this is because it's been degraded in the L morph due to the presence of this complex. If this is the case, then we would, like, endoreduplication wouldn't be happening, and we would not be elongating our filaments, resulting in our short filament phenotype. Alternatively, we do see phosphorylation of ATM1 specifically in the S morph. ATM1, it activates this SOG1. SOG1 is a negative regulator of CDKB11. That would prevent CDKB1 from forming that complex with psych, and it would prevent degradation of CDKG2 by that complex. CDKG2 actually interacts with this psych LA, and is phosphorylated at a site that was not related to degradation. In the S-morph only, we see higher phosphorylation of the psych LA in the S-morph relative to the L-morph. That might suggest that this complex is actually active. Together, this complex activates endoreduplication, resulting in filament elongation, and then resulting in our long filament phenotype. Hypothetically, this is all bioinformatics, and so we can't actually say if this is real or not. It needs empirical support, but it at least gives us a starting point. See if this hypothesis had any merit. I measured a bunch of things. I'm gonna zoom in. I'm using fancy PowerPoint. And it really made the quality of the picture go down, but I'm sure you can understand what's going on. Here we have the length of cells in the S-morph filaments, mature S-morph filaments. So this is after the flower has opened. And we have the cells of the L-morph filaments right here. And we can see that the S-morph filaments are significantly longer than that of the L-morph. Imagine there's a star there. There isn't because I am neglectful. So this might support that hypothesis that endoreduplication promotes rapid growth of the filaments of the S-morph. That might not be so surprising because if you see, we do have very rapid growth of the filaments towards that late stage. So we can see that up to a certain point, our filaments are basically the same size between the S and the L-morph. And then right before opening, we have this rapid growth of the filaments, basically over like a night. So I don't know if a night is actually that rapid, but compared to pistil elongation, it certainly seems much more rapid. Okay, so back to our little circle diagram, summary diagram. We can now throw endoreduplication up there. So hypothetically, SPH1 may be promoting filament elongation through endoreduplication. So does that mean that both of these proteins are involved in stopping mitosis from occurring? As you might think, like programmed cell death, that can be a result of premature stopping of mitosis and telling the cell that the cell needs to die. Endoreduplication, premature stopping of mitosis, telling us that the cell needs to get big. Okay, so that was cool, Paige. You did a little proteomics, but what about our receptor? What about the meat of this talk and why we all came here today? Because I'm sure this is the riveting part. Okay, so like I said, there are 77 members of SPH interna subulata, and there's SPH1. What's cool is that SPH1 evolved from a scaffold that contains, at the very least, 5 different SPHs. It looks like there might be 2 more, but they've been truncated either artificially due to the fact that this is a scaffold genome, or actually they're just actually truncated. So what we did is we actually made recombinant protein for a bunch of different SPH family members from Ternera. So we have SPH1 from Ternera subulata, SPH1 from Ternera joellii. Joellii is another distylus member of Ternera. We have TS-SPH2, 4, 3, and 5. So those are all part of that area of the genome that SPH1 duplicated from. We have SPH 25 and 29 that we synthesized, and this is because they're differentially expressed in the anther, which is cool. SPH 17 is actually expressed in the filament as well, which is why we decided to synthesize that. And then we have SPH 20 and 21, which are the closest homologs to Arabidopsis 15. So we had a bunch of different recombinant SPHs. And this was when Mike was like, you got to start working with Arabidopsis. And I was like, but why? Okay, and then I did. I just did, because sometimes you just do what your boss says. You don't question. Okay, so that's what I did. My recombinant SPHs, so I did some root assays. And so that's what I did. Basically, what I did is I took Arabidopsis seedlings, I threw them in liquid media, either containing a control, which was just buffer, or the SPH within its buffer. And we found that a bunch of SPHs actually reduce root elongation in Arabidopsis seedlings. We also did this with Arabidopsis SPHs, so Arabidopsis SPH 22, 23, and 90. And we saw a similar thing where some SPHs are actually reducing root elongation, some do not, and some kind of have an intermediate phenotype. At this point, I decided to hone in on SPH4. SPH4 had a very severe phenotype, and it's closely related to SPH1, which was my protein of interest. Unfortunately, at this point, we did not make enough SPH1 for me to really do anything with it. So I had to just say RIP to SPH1 and do my job. Okay, and so before we get like wild and like do a bunch of different things with the SPH, I needed to make sure that it was actually a result of the SPH itself and not the recombinant protein, because this SPH was attached to an FC tag. So here we just see that there is a dose-dependent effect with SPH4. Here we see that the tag does not actually reduce root elongation. It's just SPH4 that's getting the job done. And here we're just showing that structure is important for SPH function. So I digested SPH4 and then I threw it in that with some roots, and we saw nothing happened. Next, I was like, looking at actual vegetative tissue would be cool. So I set up these massive plots, and we're just going to zoom in on something that I submitted, and it is low resolution. So I'm sorry. Okay, so I set up these plots, and I treated each plant with SPH. So I literally took my pipette and I just pipetted SPH onto each plant because I'm a plant biologist. Yeah, I don't know, but that would seem like the best way to go about this without using a ton of SPH. And we see 5 days after I treated these plants with SPH, we don't really see that much of a difference. But once these plants got out to 33 days, we actually saw that the wild type plants, or the control plants, the control plants continued to grow and they look really healthy. And our SPH treated plants, really it's just like this one guy survived. The other ones, they're just dead, they're gone. Don't know if it's because they drowned and like fell under dirt, or I'm a bad plant biologist, I don't know. But that was cool because it suggests that SPH4 kind of represses vegetative growth, not only of the root, but also of the shoot. Yeah, no one brought up the patent. And I thought at this point, I'd be like, this is the basis of the patent. But I guess like Mike failed me as a boss today. Okay, cool. So now that we have this like data that showed that SPH can actually do something, SPH4 specifically, I did some co-IPs with Arabidopsis seedlings, and that like I actually did it 3 times because I'm neurotic. So I had N of 1 here, and then both of these had N of 3. So yeah, I then took these 3 data sets and I filtered all of the like proteins that pulled down with SPH4 from Arabidopsis. I removed anything that I considered a housekeeping gene, so like ribosome related things, gone, out of there. And then I came up with a filtered list of candidate interactors. Once I had this filtered list of candidate interactors, I went to the Arabidopsis Biological Resource Center, that's a typo, because I'm lazy and I'm not gonna make a bunch of knockouts myself when someone's done the work for me. And so I ordered a bunch of seeds from Ohio State's Arabidopsis Biological Resource Center, and I treated them with SPH4. Yeah, and I censored things because it's the basis of this patent that I submitted to WARF, and I knew that this was gonna be online and I didn't want WARF to come murder me. That's a joke, WARF. Please laugh, guys. We're being held hostage. Okay, anyways. I'm sorry. Okay. So what was cool is, you'll notice that we have these censored proteins that I've called AP1, AP2, AP4. They all belong to the same family, the P family. 2 of them were insensitive to SPH treatment. So that likely suggests that they are somehow involved in SPH signaling. Either they are directly interacting, which would be the strongest hypothesis considering these were identified through co-IPs, right? Or they're interacting somewhere downstream. This family is cool because it's involved in mitosis, it's involved in transport, and it binds phosphoinositides, sorry. What was also cool is that we see this protein, AS5, is also insensitive to SPH treatment. And SPH5 is cool, or S5 is cool because it's a GPI anchor protein. Which you might remember, the poppy people think there's a GPI anchor protein involved in SPH signaling. Just to make sure that this was not just like some fluke from the line that I had, I ordered additional knockout lines, and we continue to see insensitive phenotypes. So let's go back and summarize. So we've shown that PIPs are important hypothetically through our Frankenstein Arabidopsis Turner system for SPH signaling. We've shown that mitosis hypothetically is important since the P family is involved in mitosis. And we've shown that there is actually likely a GPI anchor protein that is important for SPH signaling. So it's likely actually the co-receptor, hypothetically. We also showed that SPHs are able to suppress vegetative growth. So if we look at that P family, yeah, okay. This is just an outgroup. It's not actually a member of this specific subfamily, but is part of the superfamily. So it's just there for that reason. We see that P1 is actually highly expressed throughout the plant compared to the other members. So potentially it's not that surprising that P1 was screaming at us in our data set. What's cool is P4, that protein that was interacting with SPH, that came down as a co-IP but didn't show an insensitive phenotype, it's actually highly expressed in reproductive tissue. So perhaps we're not seeing a vegetative phenotype solely because P4 plays a bigger role in the flower, specifically the pollen tube, than the vegetative tissue. Who knows? I do know that based off of other research, P4 is redundant with other members of the P family. And so hypothetically, it just might be that we have to have more knockouts, like the actual P4, P5, P3 knockout that is used in other studies. Okay, so we did our knockout root assay. That was cool. And we found this candidate interactor P. So then we made recombinant P using CHO cells. All of this has been in CHO cells so far. So then we decided we need to show that there's actual binding between these two proteins, hypothetically using, this is the Arabidopsis SPH. Okay, so the first thing I did is I made a really ugly heat map for you. So I took the recombinant SPH4 and recombinant SPH1, 2, and 3. I threw them in a tube with either recombinant P1 or recombinant P2, and I did a co-IP assay to see if P1 would pull down more, would actually pull down with these different SPHs, and if P2 would pull down with these different SPHs. SPH4 was set as the standard, so it equals 1. Everything is ratioed to it. Based off of this, you can see that SPH1 surprisingly interacts much stronger hypothetically with P1 than the other SPHs, even SPH4. SPH2 didn't really do anything at all. SPH3, it does pull down with P1, but to a much lower extent than 4. Yeah. What was cool is that with P2, we actually don't see any of the SPHs pulling down with it, which may suggest that P1 is more degenerate than P2, and it's who it's willing to perceive, I guess. We, in a collaboration with Arizona State, like he moved to somewhere in Texas, Michael Marty.
He's in Texas now, Michael Marty.
Yeah, in collaboration with Michael Marty in Texas, we did some non-denaturing mass spec. So again, we had our AP1. We went with P1 because we were able to make a large amount of P1 in CHO cells. The other Ps, we weren't able to make like a ton of it. Yeah, so this is why we were fixating just on resources alone. Okay, done talking about why we're fixating. Okay, so one thing to note with the P family is at the very beginning of the P family, they have a disordered region. And so it makes all of our graphs pretty messy like this. So yeah, you'll just have to accept this, I guess. I accepted it, but I'm not, yeah. Okay, so we see that when we combine P1 with SPH, we actually end up getting a peak that we don't see in our SPH sample, and we don't really see it in our P1 sample alone. So that kind of suggests that they are interacting. But that's really the weakest of our data, I would say, because of this, like how messy it is. And then Ben from our lab, Minkoff, that's his last name. He did some covalent labeling for me. So basically what he did is, again, he took the two proteins, he put them in a tube, and he did some covalent labeling mass spec. So this is when you are able to identify regions of the protein that are accessible to the soluble solution they're in. And we found that when we add SPH to P1, we have changes in our covalent labeling, which is pretty cool because they're occurring in that disordered region. This is a structure that I generated using AlphaFold, and we can see that it's in that disordered region. So that was cool. We have a bunch of in vitro assays. But what would be cooler is if we did the same thing, but now in Turner subulata. And so Ben, he also helped me with this. We had some Turnera stamen lysate. So we did whole stamen because it takes a lot of work to get lysate from filaments alone because they're so tiny and you need so much, and I can only have so many bushes growing in a growth chamber. So we went with the whole stamen because you have a lot more tissue to work with and it's a lot easier to get the amount of protein needed for covalent labeling. So yeah, we did some covalent labeling and we did some phosphoproteomics at the same time. And I'm gonna just show you this phylogeny real fast of the Turnera P family and the Arabidopsis P family so that you can understand what's cool and significant about this. So as you might remember, P4 is highly expressed in reproductive tissue in Arabidopsis. In Turnera, we actually have 3 P4 homologs, which suggests that maybe that like it expanded in Turner, right? And potentially maybe that's because it's important for distyly. Maybe not, who knows? Yeah, I only bring this up because TP242, we found significant differences in covalent labeling as well, specifically in that disordered region. So we can see again, here's an AlphaFold predicted structure of P42 and Turnera, and we see labeling in that disordered region. So that was pretty cool. But then it was like, what if we actually do something in vivo and not just in vitro? So then we took Turner subulata intact stamen, and we had them in solution, and we did some covalent labeling again. And what was cool is we found labeling in that same region again. We actually found much more labeling. This is still like all statistically significant. I'm only showing you the statistically significant changes. So yeah, that was exciting because it actually suggests that yes, in its native system, SPH4 is actually interacting with a member of the P family, and this P homolog, P42, might be important to reproduction, just like how the Arabidopsis 4 is only expressed really in reproductive tissue. Yeah, so that was cool. My final bit of data, it comes from an RNA-seq data set from my PhD that nobody did anything with because I was too naive with computers back then. And so I did nothing with it. So as you might remember, we have this short homocell mutant, which has short filaments. And we had RNA-seq data for the short homocell that was not published. I also had RNA-seq data for the stamen of the L-morph and the stamen of the S-morph. This was all stamen data. But what was cool is I did a co-expression analysis. And what we found is that TSSPH1, the distyly SPH, and P2.2, they're both, the expression of both of these proteins positively correlate with wild-type S-morph filaments, which potentially means, or at least supports, the potential interaction of SPH with these, this protein family, this P protein family. I want to say the name so bad, it's just hard. Okay. Yeah. And like filament elongation, so like an actual role in distyly. Hypothetically, that we'd actually have to have like transgenics to like prove this. So just to highlight the cool things that I saw, or like this data suggests that we found that GPI anchor protein that the poppy people suggest might exist. And we did support their hypothesis that PIPs are important for hypothetically SPH signaling somehow. So yeah, through this data, we supported a potential role of phosphoinositols in SPH signaling. We identified a potential co-receptor, AS5, which is a GPI-AP anchor protein that is probably required for SPH signaling. And then we identified a strong candidate receptor, but we require additional stronger support. And yeah, I've been out of the lab for like 3 months because I had a beautiful baby, like Mike showed you. Yeah. Okay, and I like to end with this quote. Most people don't know that Charles Darwin was a botanist in his later years, and his favorite thing that he studied was distyle and primrose. And you don't have to take my word because he has this quote in his autobiography. And that's cool because Charles Darwin, he made a lot of comments about how he hated himself. I hate myself. Charles Darwin, he loved distyle. I love distyle. Very relatable, very cool. And with that, I'll stop torturing you with my bad jokes, and I'll take any questions. Yay! Oh.
Thank you very much, Paige.
Yep.
So,
Those the flowers, man.
My questions. One thing I do want to add. The SPH.
Yeah.
The SPH and the P protein expression in CHO cells was done for us by Fritz Schomburg and David Rancourt, PhD student from Rick's lab and Sebastian's lab as part of their company Lytic Solutions. I'm very thankful for that. Okay, so now, any questions?
Oh, you're good. Oh,
Wow.
no.
It was close. I'll start him, Rick.
What do you think is the key piece of evidence you need to lock in your receptor?
I think I have to believe it myself. I think that's my strongest point as a scientist is that I refuse to believe anything because I refuse to think I'm competent enough to get something right. So I think there's a cool data set that Ben generated that I haven't gotten to go through, and Ben's been doing some more experiments while I've been out of the lab, and potentially those experiments will show me that I can be confident in this as an actual thing. I think that really, to prove it to myself, I would need transgenic lines of either where I'm probably like Turner. I wanted Turner, but it takes 2 years to generate transgenic Turner from experience. If someone wants to like optimize that for me, that'd be cool. But yeah, so I think transgenics.
Okay. Am I allowed to add to it?
Go,
Paige?
yeah, what would prove it to you, Mike? Mike
Well,
has the confidence. I don't.
So I think you said it perfectly well. The mass spec, the non-denaturing mass spec, that was a very noisy purple line, as you probably saw.
Yep.
We need to repeat that. We've also got some nice SEC data, which is also non-denaturing, showing interactions. We're also doing some KDs. So I think once we get a better idea of the complex, we have a pretty good idea, but like I said, that needs to be shored up. We'd like to do cryo-EM on it. Fortunately, it's just under, it's about 140 KD. So I don't know, Tim tells me it's too small. What do you say? Huh? Thank you. Well, then we're going to be over to your lab tomorrow. Rick.
So your AlphaFold predicted a lot of unstructured region in a couple of domains. I assume one of them was the EA3-2 binding domain.
Yeah.
And so you've got the other one. Have you mapped to see where this peptide is binding?
Right.
And also, I didn't say how long is the peptide?
Oh, it's approximately 140 amino acids, but then you have to subtract like, I think it's 17 for the tag itself.
So the active domain is 17.
That's the tag itself. The peptide itself is
SPH. SPH is 17. The receptor is 60 or 70. what you see here is 554 amino acid long.
Yeah.
So Rick, you're asking, this is the phosphoninositide binding region that's known. This is the unstructured region that was predicted to have a ligand, but they didn't know what it was. We believe now we know what that ligand is, SPH.
Is that in your alpha-fold at all?
In the alpha fold, yeah,
Either like these,
this is the unstructured
Yeah.
region. This is the
I have like
phosphoninositide stuff.
this. You can see it's up here too.
That's all this. Any other, oh, do you need this? Oh, yeah, actually, I'm sorry, I should have done this.
Really cool talk. So you're studying a peptide hormone that's primarily involved in flower morphology in Ternera. Why do you think it's causing those growth defects in Arabidopsis in the root and vegetative tissue?
Well, somehow you have to stop vegetative tissue from growing so you can start reproducing, right? And so hypothetically, there's always the possibility that it's involved in stopping vegetative growth so that we can begin actual reproductive growth, right? We only know the function of 2 of the family members, and the poppy one clearly is an exceptional case of neofunctionalization, given that its receptor is papaverase specific.
See that word neofunctionalization?
Yeah.
It's talking about the matrix.
Oh, God.
Turns out.
So, yeah.
Explain to me why only poppy has a sequence. This sequence is not in any other species. Period.
Yeah,
It's
unfortunately.
really amazing. And it's probably a calcium channel. It's a very small transmembrane protein. Slimy.
Did I answer the question sufficiently? Okay. Cool.
Oh,
Any other questions? Well, again, let's thank Paige for